In the cell, there are many methods for controlling how DNA is read and expressed. Several of these methods, called epigenetic modifications, can turn specific genes on or off without altering the DNA sequence. One of these methods is DNA methylation, which occurs when a methyl group is added to a DNA base. When this happens, additional proteins can no longer bind to this region of DNA. This prevents the DNA from being transcribed into RNA. The gene is turned off. Normal methylation patterns are important for the development and function of healthy cells. In cancer cells, abnormal DNA methylation patterns are seen. Genes that would normally suppress tumor growth see higher levels of methylation and are turned off. At the same time, genes that increase tumor growth, called oncogenes, see a decrease in methylation and are turned on. This results in unregulated tumor growth.